to Intro 101. My name is Chris, and I am your neuroscience and psychology graduate student assistant. I'm here to assist you with the related science basics that you interact with on a daily basis. My goal for doing this is to help you start or continue to build a foundation in human biology and physiology that can better help you engage with the information you consume from your favorite health and wellness educators and influencers. Much like the way a graduate assistant helps prepare intro students so they can better engage with the lectures delivered by their professors. Or maybe you're here today because last episode I dragged you through Middle Earth and left you hanging off a cliff and you just want to land in a soft, fluffy pile of knowledge. Either way, stick with me for the next lots of minutes and I'll do my best to rescue you. But I can't guarantee a soft or fluffy landing. The gamete journey may have ended in the last episode, but the baby you journey is just beginning, and I can't even find adequate words to describe how mind-boggling it is that we can grow humans inside of our bodies, and that they also kind of grow themselves. When you were only a few cells old, you plugged into your mom or parents' life force and got down to business. And I'm going to tell you about some of that business. Today's episode is Intro to Neurodevelopment 101 Part 3. Prenatal neurodevelopment. It is entirely devoted to the moment you became a bunch of rapidly dividing cells, and will follow those cells up to where the nervous system kicks off, beginning with the development of the neural tube, and then follow brain development up to the moment you entered this world. As I alluded to in the last couple episodes, human development is dynamic and nuanced. We are beings with layers on top of layers of complexity to our existence. So, as I said in the previous episodes, I will be sticking as closely to basic biology and homeostatic processes as possible. Though again, we will look at what can happen when the different stages of prenatal development veer away from a textbook typical developmental trajectory, and how that can potentially lead to atypical outcomes. But please remember, we don't yet fully understand the etiology of many atypical outcomes, or all of the biological markers involved in nervous system development disorders, including those that lead to neurodiversity. So please, do not use the information presented in this or any of the episodes to self or other diagnose. If you have concerns regarding prenatal developmental milestones, please speak with a medical professional. I'll post any helpful links I know of in the show notes, but if you're aware of others, do let us know in the comments below. Before we start, I'd like to respectfully acknowledge that Intro 101 is recorded on the unceded and ancestral territories of the Kwantlen, Katzie, Matsqui, and Semiahmoo First Nations. Additionally, we at Intro 101 believe wholeheartedly that science is for everybody. In fact, the richness, complexity, and strength of scientific research is astronomically improved by the inclusion of many varied voices. To ensure everybody has access to education, research resources, and representation across scientific fields the world over, I will be sharing organizations in the show notes that provide access to STEM education and research resources, and I ask that you check them out and support them in any way that you can. If you already advocate or generate opportunities to provide equitable access to STEM education and resources within your own communities, or support other STEM education organizations not listed here, please share them in the comments below if you'd like others to contribute as they are able. All right, welcome to class. Thanks for being here. Let's crack on with Intro to Neurodevelopment 101, Part 3, Prenatal Development. First, I'd like to mention, if you've just arrived at this channel through this episode, this series is an episodic integration of information that is building a bigger picture of the nervous system, its components, its physiology, and its collaboration with other body systems to keep your body in homeostasis. If you're new to homeostasis and/or the nervous system, or a little rusty on how it works, it may benefit you to view the previous episodes in this series first. A general understanding of what homeostasis is, nervous system function, general cell physiology, neuron and glial cell function and physiology, gametogenesis, and fertilization will help make sense of this episode, all of which are covered in the first 7 episodes of this series. Okay, so let's do a quick recap from the last couple episodes. We learned about gonads, which are testes and ovaries, and how they produce gametes. Gametes are the reproductive cells responsible for creating life. Testes produce sperm cells, and ovaries produce oocytes, also called egg cells. both develop under very different processes and on very different timelines. We looked at the shocking ways bodies select for which gametes get a chance at fertilization, including the way oocytes may volunteer themselves for recruitment, could also partially volunteer their demise if not selected. And the selected oocyte has to wiggle out of the follicle and make their way to freedom that only lasts about 24 hours. And if it doesn't unite with a sperm cell in that time, what's left of its former follicle will wreak bloody vengeance on the uterus. Equally intense is the death-defying journey sperm cells have to take in order to make it to a fallopian tube to fertilize an oocyte. Their selection process is at the mercy of acid armor, immunoshields, hydrodynamics, Keanu Reeves-level speed strategies, and a chemical serenade by the oocyte akin to the mythical siren. Because let's be honest, though the winning sperm cell gets to drop its chromosomal wad into the oocyte, the rest of it has to die. But the oocyte, well, this is where we left off in the last episode, the brink of fertilization. Now, inside the oocyte, molecular fireworks kick off. Because if you remember, the oocyte is still in an arrested state of second meiosis. And the fusion of the sperm cell to the oocyte activates the oocyte. And a bunch of things happen in a short span of time. As I said, the fusion triggers molecular events that prevent other sperm from fusing with the oocyte. And while it does so, it also removes factors responsible for meiotic arrest. So the oocyte can resume meiosis. The oocyte completes meiosis, and it's now a mature haploid ovum and a small polar body that will eventually degrade. The sperm cell has also deposited its haploid nucleus into the cytoplasm of the ovum, and as I've alluded to already, its tail and mitochondria fall away and degenerate. The ovum becomes a single-cell zygote with 2 haploid nuclei, which are now called pronuclei. The pronuclei migrate toward each other and begin to replicate their DNA, and when they meet, the membranes between them dissolve and they fuse together. Then, both half sets of chromosomes fuse to make a whole complete set. The cell then undergoes its first mitotic division, and there are now 2 daughter cells. This first mitotic division is complete about 30 hours after the zygote formed. It's still in the fallopian tube, but has started its migration towards the uterus, and it will slowly make its way to the uterus over the next 5-ish days. As it does, it undergoes more mitotic division, also called cleavage. So while it's migrating through the fallopian tube, the 2 cells cleave into 4 cells. Then those cleave into 8 cells, and then 16 cells. And this 16-cell mass is called the morula. It's possible that the fallopian tube secretes all kinds of growth factors and other molecules that support zygote development, and that might be why it takes as long as it does to get to the uterus, because it needs time to synthesize these factors and benefit from them. By the time it reaches the uterotubal junction, that 16-cell morula has divided into a 32-cell blastocyst. The blastocyst has an inner cell mass and fluid, surrounded by an outer cell mass, and surrounding that is the zona pellucida. It's still there. It won't stay much longer, but it likely remains attached up to this point for a few reasons. One is to protect the cells during cleavage. It keeps them all together. Otherwise, studies have shown that without the zona pellucida, the cells that initially divide after fertilization just fall apart. Another reason the zona pellucida sticks around is to prevent the blastocyst from implanting in the fallopian tube, which would lead to a tubal pregnancy, also called ectopic pregnancy. And those can be incredibly dangerous to the parent and have been cited as the leading cause of first trimester mortality if left unchecked. It is treatable, though, and can sometimes even resolve itself. Another reason why the zona pellucida remains attached to the blastocyst is it may provide immunological protections in the uterus until the blastocyst can attach to the uterine wall. It would only be for a few days, but I suppose given the fragility of this tiny clump of cells, any protections the zona can offer just increase the chances of successful implantation. I'm curious to know if the zona pellucida itself has some kind of baby on board tag that signals to the uterine immunological response to please don't attack, but I'm not sure. And as we'll see in a minute, the immunological response of the uterus may contribute to an important event that helps the blastocyst shed the zona pellucida. Okay, we're going to pause here for a second because I'd like to introduce you to some vocabulary that will pop up a lot during the rest of this episode. You're going to hear the word differentiation a lot. I'll also mention pluripotent stem cells and multipotent stem cells because they are directly related to differentiation. So differentiation is when stem cells grow and mature and form a specific type of cell that has a particular function. There are different kinds of stem cells. Some can differentiate into any kind of body cell, and some can only differentiate within their cell lineage. Pluripotent stem cells are the kind that can differentiate into any cell in the human body. They can self-renew and differentiate into any kind of brain cell or bone cell, muscle cell, or even a blood cell. There are no limits to proliferation and differentiation of pluripotent stem cells. There are a few other kinds of stem cells, and one of them is called a multipotent stem cell. And those are the stem cells that have more limitations than pluripotent stem cells. Multipotent stem cells can be found in any type of tissue, but their differentiation is restricted to that particular tissue or cell lineage. So a multipotent blood stem cell can only differentiate into a red or white blood cell or a platelet. We'll see both pluripotent and multipotent stem cells do their business in this episode. And I wanted to mention them and the process of differentiation so when you hear those words, you have them in your head already. Ah yes, differentiation. The cell is growing up to be a neuron or epithelial cell, or whatever kind of cell it has the capacity to become, depending on what kind of stem cell it is differentiating from. Okay, back to our blastocyst. As it's migrating into the uterus for implantation, its cells have been differentiating into specialized cells that will perform more functions. The inner cell mass cells have differentiated into the embryoblast, and those cells are now embryonic pluripotent stem cells. So they will have the capacity to differentiate into either more pluripotent stem cells, or they're going to differentiate into cells that will become tissue. And those tissues will become organs, and those organs will become body systems. But right now, they are embryonic pluripotent stem cells just waiting to become a whole new body. The outer cell mass is differentiated into trophoblast cells, and it is composed of multipotent stem cells, so they're going to differentiate into other trophoblast cell subtypes. One subtype is cytotrophoblast cells, and the other subtype is syncytiotrophoblast cells. And these cells won't differentiate beyond this, but they have a very important job, and that is to facilitate the production of the fetal placenta. But more on that in a minute. So, the blastocyst has made its way out of the fallopian tube, it's differentiating its cells, and it's still surrounded by the zona pellucida, which so far has been protecting it. But it must shed the zona pellucida in order to initiate implantation. The process is called zona hatching. Yeah, the blastocyst actually hatches out of the zona pellucida. The hatching process involves pressure from the fluid accumulating inside the blastocyst and causing its expansion, which thins out the zona pellucida on the outside. Simultaneously, signaling cascades between the uterus and blastocyst that involve growth factors, enzymes, and possibly pro- and anti-inflammatory cytokines begin to degrade the zona pellucida. The combination of zona thinning because of blastocyst expansion and the enzymatic weakening of the zona pellucida, these together culminate in the cracking and shedding of the zona shell. There seems to be a lot of crosstalk between the uterus and the blastocyst at this stage. So probably noteworthy that if any part of those signaling cascades fail and the zona hatching is delayed or doesn't happen, the blastocyst likely won't be able to adhere to the uterine wall, and adherence is critical for implantation and successful pregnancy. If you remember from the last episode, the uterine lining has been preparing for this blastocyst since ovulation, and under progesterone regulation, it's become thick and cushy and sticky and ready to cuddle the blastocyst. And by cuddle, I mean allow the blastocyst to burrow into its lining. But it has to hatch first, so adhesion molecules in the trophoblast cells can connect to the adhesion molecules in the endometrial cells in the lining of the uterine wall. But how does it know where to adhere for implantation? Well, one analogy I came across compared blastocyst adhesion to tossing a tennis ball down a hill thickly coated in molasses or treacle, slowly rolling to a stop, and where it lands is where it implants. So you can imagine the fallopian tube using smooth muscle contractions and fluid flow nudges the blastocyst out into the uterus. It's probably not shooting it out, though I kind of wish it did, like a straw in a spitball. But it doesn't, probably, because it actually takes days to move a couple of centimeters from the fallopian tube opening to the implantation site. And zona hatching itself takes hours. But as far as its trajectory, the blastocyst seems like it just sort of slowly falls out of the fallopian tube, hatches along the way, makes contact with the uterine wall, and slowly rolls along the wall until it can't anymore. The combination of sticky mucus and the presence of adhesion molecules on both the blastocyst trophoblast cells and the endometrial cells in the lining of the uterus will eventually slow the blastocyst's roll to a stop and initiate adhesion and implantation. The exact timing of this is understood to start at day 3 and end at day 7 after fertilization. So around day 8, the blastocyst has attached to the uterine wall, and the process of implantation commences. So the outer trophoblast layer has differentiated into cytotrophoblast cells, and they start proliferating at the side of the blastocyst that is attached to the uterine wall. As they proliferate, the new cells work their way into the uterine wall layers. Here's where it takes an unusual turn. The new cells also lose their membranes. Yeah, these proliferated cytotrophoblast membranes rupture and disintegrate, and their contents, which is cytoplasm and nuclei, spill out, and all of those loose nuclei and cytoplasm will gather together and form a syncytium. A syncytium is exactly that. It's a cytoplasmic mass with loads of nuclei. And this mass protrudes and spreads into the uterine wall, and this becomes the syncytiotrophoblast. So just for clarity, the outer layer of the blastocyst still has cytotrophoblast cells that have remained inside their plasma membranes, forming the blastocyst structure and adhering to the uterine lining. It's just the cells that proliferated after adhesion that have formed the multi-cytoplasmic nucleic protrusions called the syncytiotrophoblast. The syncytiotrophoblast's job is to properly embed the blastocyst into the uterine lining in order to establish and facilitate nutrient transfer and hormonal, metabolic, and immunological function to the blastocyst. The syncytiotrophoblast is working its way deeper and deeper into the uterine wall, and to do this, it releases enzymes that are degrading cell adhesion molecules, so it can dislodge endometrial cells of the uterus. Cell adhesion molecules do what it says on the tin. They hold cells together. So the syncytiotrophoblast is dissolving proteins that hold uterine cells together to dislodge them, so that it can expand into the uterine lining and make room to embed the blastocyst completely into the wall. As the syncytiotrophoblast works its way deeper into the lining, it pulls the blastocyst deeper into the wall with it. But that's not all. It's also looking for uterine blood vessels, and when it finds them, it will rupture them to establish a connection between parental blood flow and the syncytium. And this is the beginning of placentation. It's the beginning of nutrient transfer and hormonal, metabolic, and immunological function between parent and embryo. By about day 10, the blastocyst is fully burrowed into the uterine wall, and it's surrounded and covered by endometrial cells. Important to note, while pre-implantation and implantation is happening, the uterus is secreting immunosuppressive factors so it doesn't reject the blastocyst as an invasive parasite. The syncytiotrophoblast is also releasing an immunosuppressant called human chorionic gonadotropin, or HCG, which also has another job, and that is to signal to the corpus luteum. Remember, that's the former follicle shell turned tiny cronut pumping out progesterone in the ovary. So HCG signals to the corpus luteum that this pregnancy is a go. And please, do not tell the uterus to shed its endometrial lining. That endometrial lining is going to supply all kinds of nutrients to the embryo, so it is much needed. So the corpus luteum doesn't die off just yet, and will continue to pump out progesterone until about the 12th week, which is when the placenta is established and can take over hormone supply. And, as mentioned, nutrient supply and shared immunological and metabolic function. Okay, at the same time the trophoblast is doing all of its cytotrophoblast and syncytiotrophoblast differentiation and implantation business, the embryoblast cells differentiate into two different layers of more pluripotent stem cells called the epiblast and the hypoblast. And together they form something called the bilaminar disc. And this is the beginning of the embryo. As the hypoblast continues to develop, it forms a cavity that will become the yolk sac, and this will help the embryo develop red blood cells later on, and it will also serve as nutrient and gas exchange with the uterus until the placenta is formed later on. Above the epiblast layer, a fluid-filled amniotic cavity is forming. By about day 15, more morphological changes happen to the epiblast layer. These changes are called gastrulation. And what happens is the cells along the midline and caudal, or tail end, of the bilaminar disc begin to thicken and create an indentation called the primitive streak. There's also a circular indentation that develops at the cranial end, and that's called the precordial plate. It will eventually develop into the buccopharyngeal membrane. And at the caudal end, another plate will eventually develop into the cloaca membrane. This is the beginning of your body's major axes. The buccopharyngeal membrane at the cranial end will eventually become your mouth hole, and the cloacal membrane at the caudal end will become your bum hole. At the cranial end of the primitive streak, cells start dying off to form a circular depression called the primitive node, or pit. And cells in the middle of the primitive streak also start dying off to form a depression that runs the length of the streak towards the caudal end. This long depression is called the primitive groove. The epiblast cells along the perimeter of this primitive groove start releasing growth factors that trigger the epiblast cells on either side of the groove to let go of each other. This allows those epiblast cells to migrate inward into the groove and move beneath it. Then they migrate from the caudal end to the cranial end, invading the hypoblast layer and differentiating into endoderm cells. And they eventually replace the entire hypoblast layer. Other epiblast cells migrate and form a layer in between what were the hypoblast and epiblast layers to form a layer of germ cells called the mesoderm. What remains of the epiblast cells is now called the ectoderm. When there are 3 distinct layers of cells and they have completed formation, it marks the end of gastrulation. So gastrulation is when the bilaminar disc has turned into a trilaminar embryonic disc. And again, those 3 layers of germ cells are called the endoderm, mesoderm, and ectoderm. And these germ cells are now multipotent stem cells that will differentiate into cells of the specific lineage each germ layer constitutes. So the endoderm cells are the precursor cells to mostly digestive and respiratory derivatives. The mesoderm cells are precursor to connective, urinary, and bone derivatives. And the ectoderm gives rise to the nervous system, as well as some skin derivatives. One small note, there are parts of the mesoderm layer where there are no mesoderm cells. One is where the notochord will develop, more on that in a minute, and the other areas are where the precordial plate and the cloacal plate are located. So we're roughly at about week 3 of development, and this next major developmental event is called neurulation, and it is a pivotal moment for this series. We are about to embark on the development of the nervous system. From this point on, I'm going to mostly focus on the nervous system. I mean, it's kind of the main event at first anyway, but as embryonic, then fetal development progresses, there are loads of other tissues and organs and body systems that undergo their development simultaneously. But we're going to mainly focus on the nervous system, specifically the formation of neurons and glia. Other body systems will come into play, but only in the ways they contribute to nervous system formation. There are also other physiological changes happening between the uterus and the embryo, like placenta, umbilical cord, and amniotic fluid development, and we won't cover that in this episode either. I hope the details of implantation were enough to give you an idea of how the embryo and uterus initially connect in order to transfer nutrients to and waste from the embryo. Of course, as the embryo develops, so will an umbilical cord, and that will become the main transfer mechanism between baby and placenta. Okay, so we left off with the trilaminar embryonic disc. We still have the primitive groove and the primitive pit. So what happens next is the top layer of germ cells, the ectoderm cells, start releasing more growth factors that allow for migration of some of those cells down into the primitive pit and into the mesoderm layer between the ectoderm and the endoderm, where there aren't any mesoderm cells. And they'll migrate forward toward the cranial end and the precordial plate. Between the mesoderm cells of the mesoderm layer and the precordial plate, these migrating ectoderm cells will form a tubular process called a notochord. And the notochord is super important because it's going to kick off neurulation. The notochord releases lots of different growth factors that signal to the ectoderm cells to start making loads more of themselves. So as they proliferate over and over, the ectoderm layer thickens into what is now called the neural plate. And as the cells proliferate, a disproportionate number accumulate at the lateral edges of the neural plate, elevating the plate to form neural folds. As it does this, it creates a space in between the neural folds called the neural groove. And along this neural groove, this fascinating physiological event happens called invagination. And in this instance, it's like the neural groove is being pulled down from the cranial to caudal end, forcing the neural folds to come together at the top. The neural folds will fuse together, beginning in the middle of the embryo, and then fuse toward the cranial and caudal ends, converting the neural groove into a neural tube positioned underneath the ectoderm. The neural tube will go on to develop the central nervous system. Now, during neural fold fusion and neural tube conversion, some of the cells on the crest of the neural tube detach and form a new cell population called the neural crest, and those will go on to form the peripheral and enteric nervous systems. At this stage, both the cranial and caudal end of the neural tube are still open. The cranial opening of the neural tube is called the anterior neuropore, and the caudal opening of the neural tube is called the posterior neuropore. And I just want to pause here for a second because these ends of the neural tube are not meant to remain open. They are meant to eventually close fairly soon after neural tube development. But why I want to draw your attention to these ends is because if either or both neuropores fail to close, it could lead to neural tube defects. And in order for these neuropores to achieve closure, they need to undergo intense, complicated processes of nucleotide biosynthesis and methylation. Nucleotide biosynthesis is involved in DNA replication, and methylation is involved in DNA repair, protein function, and gene expression. So both of these neuropores require these complex processes to achieve closure. But here's the kicker. In order for these processes to do their business, they need a very particular molecule called folate. Folate provides specific derivatives that fuel these processes, and they are needed in abundance. So if you're wondering why doctors prescribe folic acid, especially for the first trimester of pregnancy, this is why. If those neuropores do not have the folate resources they need to achieve closure, the nervous system will not develop properly. And depending on the neural tube defect, that could be fatal. Anencephaly is an example of a neural tube defect that resulted from the failure of anterior neuropore closure. It is characterized by partial or complete absence of brain and cranial development, and it is incompatible with sustained life. Spina bifida is a neural tube defect that results from the failure of posterior neuropore closure. It's characterized by spinal malformation. There are a number of types of spina bifida. Most or all are compatible with life, but can have varying levels of symptom severity and treatment. Now, there are a number of different types of neural tube defects. Not all are the result of folate deficiency, but also noteworthy, folate could be important fuel for other neurulation processes leading up to neural tube formation. And even before that, which adds some context to how soon one should begin folic acid supplementation. Neurulation takes place in weeks 3 and 4 of gestation, so it's feasible it's happening before someone would even know that they're pregnant. So if you're planning for pregnancy, it may be worth speaking to your doctor about preemptive supplementation. I'm not familiar with dosage or how long it stays in your system, and you may also have a folate-rich diet, which may minimize some of the risk of deficiency during early embryogenesis. There's a lot of factors to consider. I just thought it was interesting to know just how important folate is to the developing embryo. I mean, I knew it was important, but I didn't appreciate just how many incredibly complex molecular processes relied so heavily on it to be able to perform their functions. Okay, let's keep going. If the body's supply of folate is ACEs, at some point during week 4, the anterior neuropore will close, and a few days later, the posterior neuropore will close. And now we have this complete neural tube. And also, of course, we can't forget those neural crest cells beside the neural tube. The cells inside the neural tube have differentiated into neuroepithelial stem cells, and they will continue to proliferate and differentiate, and we'll get into what's happening there in more detail in a minute. But let's take a quick peek at what's happening from a broader perspective. As the cells proliferate, again, at slightly disproportionate rates along the tube, there is a swelling at the cranial end, and then more swelling underneath that. And this is the neural tube morphing into vesicles in a process called vesiculation. And these first 3 vesicles are called primary vesicles. And in cranial to caudal order, there is the forebrain, also called the prosencephalon, the midbrain or mesencephalon, and the hindbrain or rhombencephalon. And below that is the beginning of the spinal cord. By about the 5th week of development, as the cells continue to proliferate and differentiate, the primary vesicles subdivide into 5 secondary vesicles. So the forebrain has subdivided into the telencephalon and diencephalon. The midbrain stays the same, it remains the mesencephalon. And the hindbrain subdivides into the metencephalon and myencephalon. The telencephalon will eventually become the cerebrum, which is most of your brain. And by adulthood, we'll have fully developed lobes, and they are the frontal, parietal, temporal, and occipital lobes. And we'll get deep into their functions as they develop through childhood and into adulthood in future episodes. The diencephalon will become the thalamus, hypothalamus, posterior pituitary, and epithalamus. The epithalamus includes a bunch of bits we haven't mentioned yet in this series, but we will get to those later on. The diencephalon will also give rise to the retinas, but at this stage they are just 2 eye cups. The mesencephalon, or midbrain, will give rise to the cerebral peduncles, which sounds like a card game you play with your gran on a rainy afternoon. But they are actually 2 significant stalk-like structures with loads of sensory and motor nerve fibers connecting the cerebrum to the brainstem. The mesencephalon will also give rise to the tegmentum. You may recognize this word already if you remember from the neuron physiology episode. The ventral tegmental area, or VTA, is part of the tegmentum, and this area is home to a cluster of dopamine neurons which kick off the mesocortical dopaminergic pathway. The tectum is the other structure the mesencephalon gives rise to, and it contains 2 structures, the superior and inferior colliculi. The superior colliculus is involved with visual reflexes like tracking objects, and the inferior colliculus is the main auditory input hub where sound recognition happens. The 4th secondary brain vesicle, the metencephalon, will give rise to the pons and cerebellum. The pons is involved in many functions. It has clusters of cranial nerves that control involuntary respiratory functions, as well as facial, sensory, and motor reflexes. It also has clusters of neurons that participate in sleep, respiratory, urinary, auditory, gustatory, and vestibular functions. The cerebellum is involved in motor coordination and movement, among many other things. The 5th secondary vesicle, the myencephalon, gives rise to the medulla oblongata, and that connects the brainstem to the spinal cord, and is also very much involved in motor and respiratory function. So this is a lateral view of an embryo at about 5 weeks. But let's take a look inside these morphing vesicles. Because cells are rapidly dividing and differentiating. And how do they know where to go and what kind of nervous system cell to become? Because remember, there are different types of neurons and many kinds of glia. And also remember, at birth, the brain has twice as many neurons as it will have in adulthood. So we're looking at a 32-week time frame to pump out possibly 200 billion neurons alone, not including glia, and of course, not including the trillions of other cells that will make up your innards and outards. It is phenomenal to think about the multitude of molecular and cellular functions knitting together this mighty wee embryo. But let's focus on the central nervous system because it is wildly coordinated and strategic in its development. We're going to specifically look at the birth and differentiation of neurons in a process called neurogenesis. You have most likely come across this term before. The body goes through multiple phases of neurogenesis up until adulthood. Neurogenesis may continue into adulthood in humans in some structures, but we'll tackle that in a future episode. During embryogenesis, neurogenesis is in full swing, and this is what's happening. Let's rewind a titch to the completion of the neural tube and neural crest cells. As the neural tube is vesiculating, it forms 3 cellular layers. The inner layer is called the ventricular zone, the middle layer is called the intermediate zone, and the outer layer is called the marginal zone. Both the central and peripheral nervous systems will emerge from these zones. We're going to spend the majority of the time looking at central nervous system brain development, but I'll first quickly explain how spinal cord formation kicks off. So inside the neural tube we have these 3 zones, and the ventricular zone is replete with neuroepithelial stem cells, and they will continuously divide and multiply. And at the caudal end of the neural tube, that is where the spinal cord will begin to develop from these proliferating neuroepithelial stem cells. In the intermediate zone, these cells will differentiate into cells called neuroblasts, and those will start migrating to the marginal zone and beyond, and there they will form structures. The upper or dorsal structures they form are called the alar plates, and the lower or ventral structures they form are called the basal plates, and those mature and develop throughout embryogenesis. The alar plate will become the dorsal gray horn, and the basal plates will form the ventral gray horn. The neurons that form the dorsal gray horn will mature into sensory neurons, and the neurons that form the ventral gray horn will mature into motor neurons. And the tube that remains in the middle is where the cerebral spinal fluid will live. But we'll spend more time on the spinal cord in a future episode. Okay, let's look more closely at brain formation, specifically the cerebral cortex. The cerebral cortex makes up more than 80% of your brain mass, but interestingly, only 19% of your neuron count. Still, the development of the cerebral cortex is a fascinating process and indicative of neurogenesis in other brain areas. So we begin in the telencephalon because these cells will give rise to the cerebrum. Within the telencephalon, we have our ventricular zone. And as I said, it is chock full of neuroepithelial stem cells, and they will continuously divide and multiply, and as they're proliferating, they will differentiate into radial glia cells. All cortical neurons and glia are born from radial glia cells. I briefly mentioned them in the episode on glia physiology, but I'll get into them more now because neurogenesis is where they shine. So as radial glia divide, some will differentiate into either neuroblasts or glioblasts, and those are neuron and glia precursors. And some will divide into more radial glia cells and stay in the ventricular zone. And some radial glia will provide this amazing scaffold for developing neurons and glia to use as a guide to migrate towards their destination, where they will differentiate into their final neuron or glia form. So let's follow one radial glia progenitor cell through the process of neurogenesis to its final destination, which in this example is going to be a pyramidal neuron, and its home will be in the cerebral cortex. So let's go back to our zone layers because this provides a map for our neuron's trajectory. If we take a cross-section of the telencephalon, you can see 2 cavities on either side, and those will give rise to the lateral ventricles in the developed brain. There are 4 ventricles in the brain. The first two are the lateral ventricles in each cerebral hemisphere. The third is part of the diencephalon, and the fourth is in the midbrain. Okay, so we have our embryonic telencephalon with its baby lateral ventricles and the ventricular zone, where the radial glia progenitors are revved up and ready to go. That is the layer of cells closest to the lateral ventricles. Makes sense, right? Ventricular zone above the ventricles. The next layer is the intermediate zone, and it's in the middle, and then the marginal zone is at the top. As a point of reference, we'll call that the peel surface. This is because in adults, 3 layers of membranes called meninges will form between the cortex and the skull, and they cushion the brain from the skull and have other functions that we'll get into in a future episode. The 3 meninges layers from the skull to the cortex are the dura mater, the arachnoid, and the pia mater. The dura mater is derived from the mesoderm, but the arachnoid and pia mater are derived from the ectoderm, like the rest of the nervous system. The underside of the pia mater adheres to the cortex, so the very outer layer of what will be the cerebral cortex, that will be covered with pia mater, and we call it the peel surface. So that's why we'll use the term peel surface as a reference so you can have context to where our example baby neuron is migrating. It is migrating toward the peel surface. Okay, so at around week 4 or 5 of gestation, the radial glia progenitor cells in the ventricular zone are proliferating like mad. And some radial glia, with their cell bodies in the ventricular zone, will form unipolar processes that extend all the way to the peel surface. At the same time, the meninges are beginning to form, and it's possible even in this primitive stage of development, the meninges express factors that help direct neuronal development and migration. But around this time, some of the radial glia progenitor cells shift the way they proliferate. Up to this point, their mitotic division has had a vertical cleavage orientation, and that produced 2 symmetrical daughter cells that went on to divide and multiply into more radial glia cells. Now, they will add a horizontal cleavage orientation to their mitotic division, and this will produce 2 asymmetrical daughter cells. So now, as the radial glia progenitor cells go through mitosis, they will divide either horizontally or vertically. And to clarify, I mean the plane of cleavage will be either parallel or perpendicular to the ventricle layer. But why are they doing this? Because this will determine each of the daughter cells' fate. Yes, fate. We are back to fate. If you don't know what I'm talking about, go back to the glia episode and listen to how Schwann cells find their axon fate. It's a love story for the ages. But radial glia progenitor daughter cell fate is less of a love story and more of a destiny, fate, hero's journey story. And this is what happens. If the radial glia progenitor cells divide horizontally, the daughter cell that is facing the peel surface side will go on to differentiate into a neuron or glia cell. The daughter cell facing the ventricular surface will remain a progenitor cell in the intermediate zone. The cells that divide vertically, both of those daughter cells will remain as radial glia progenitor cells and divide again in the ventricular zone. But how does the cell know the difference? How does the daughter cell know it's symmetrically divided or asymmetrically divided? More specifically, how does the peel-facing horizontally cleavage daughter cell know it's supposed to begin migrating and become a neuron? Well, it's possible that special proteins called transcription factors tell the daughter cells where to go. And they do this by expressing themselves in an asymmetrical pattern. So when we look at the radial glia progenitor cell, there is a particular transcription factor that is expressed toward the ventricular surface, and that factor is called NUM. And another transcription factor is expressed toward the peel surface, and that factor is called Notch1. So when the cell divides vertically, the daughter cells have a symmetrical expression pattern of both of these factors. They will have both ventricular surface expressing NUM and peel surface expressing Notch1. And if they have both of these factors expressing at those particular poles of the cells, that signals to the cells to remain in the ventricular zone and go back into the progenitor cell cycle for more mitotic division. But when the cell is horizontally divided, the daughter cells have an asymmetrical expression pattern of the two factors. The peel surface facing daughter cell will have higher Notch1 expression and virtually no num expression, and the ventricular surface facing daughter cell will have a higher num expression and little to no Notch1 expression. So what does all of this mean? Well, it seems that by removing or cleaving num transcription factor from the cell and leaving Notch1 transcription factor to express itself, this could restrict which molecular cues the cell can receive. It's also possible that Notch1 expression activates genetic mechanisms that kick off neuron or glia development. So the cell with only Notch1 follows its signaling lead down the path of development because there's not enough of any other transcription factor to tell it to do otherwise. Likewise, the other daughter cell with only num transcription factor expression remains a progenitor cell and migrates to the intermediate zone. And when enough of those intermediate progenitor cells accumulate in the intermediate zone, they form another zone layer called the subventricular zone, and it sits on top of the ventricular zone. And those intermediate progenitor cells in the subventricular zone will symmetrically divide and eventually produce post-mitotic daughter neurons. There are a number of studies that look at other mitotic cleavage orientations that aren't exactly vertical or horizontal, but they're using mostly fruit fly, zebrafish, and rodent models, and they have slightly different outcomes than I just described. And there may be other transcription factors that work independently or in cahoots with Notch1 and num to direct the daughter cell fates. It's well beyond introductory level instruction, but I have posted a couple papers in the references section of the show notes that might interest you. So we have radial glial cells with their cell bodies in the ventricular zone and their unipolar radial fibers or processes that extend to the peel surface. We also have intermediate progenitor cells accumulating and forming the subventricular layer. And we have Notch1 daughter cells ready to migrate as neuroblasts or glioblasts. So let's follow the neuroblast from here. It is ready to migrate. And in the telencephalon, cells will migrate in a specific way, and that way is called radial migration. And that is when cells migrate directly away from the ventricular zone in a fairly straight path towards the peel surface. Cells migrating from other parts of the neural tube will migrate tangentially, which is movement that is parallel to the ventricular surface. The cell we're following will migrate radially from the ventricular surface toward the peel surface. And for the most part, neurons that develop from the telencephalon and migrate radially will become pyramidal neurons. The glia cells that migrate radially from the telencephalon will become astrocytes. Okay, so this next part is kind of the best part of neurogenesis, at least for cortical neurons. Partly because it's so clever, but also slightly comical. So we have a radially migrating neuroblast. It finds a radial glia cell that has that unipolar process extending to the peel surface. And it uses the process as a guide to migrate toward the peel surface, but not so much like a railing as you might expect. The neuroblast kind of wraps itself around it and hikes itself along it, like Bridget Jones trying to climb back up a fireman's pole. Less clumsy, but no less determined, and likely with the help of cell adhesion molecules. Each cortical neuroblast will travel this way until it reaches the cortical layer it's going to form. So let's pause here for a second to look at the layers of neurons that will comprise the cerebral cortex and how those layers form. The developed cerebral cortex has 6 stacked layers of neurons and glia and some unmyelinated axons, all of which make up the gray matter of the brain. The 6 layers together is about 2 to 4 millimeters thick. Each layer is specifically arranged with distinct populations of neuron types that have different genetic, structural, and functional capacities that together increase the computational efficiency of the brain. During brain development, these layers form in a very particular and organized manner, which is inside out or bottom up depending on how you approach it. The sixth layer, which is the furthest from the peel surface, forms first. There are likely factors the neuroblast is expressing along the way that tell it where to stop to form that layer. But nonetheless, the first migrations will form that sixth layer first. And once those cells are in place, the fifth layer will form. And when those cells are in place, the fourth layer will form, and so on, until all 6 layers are formed. Pyramidal neurons are dispersed throughout layers 2 to 6. Layer 1 doesn't actually have cell bodies, it's mostly dendrites and axons from the other layers. So our neuroblast is Bridget Jonesing it up a radial glia process, and it will stop at layer 5 for a couple of reasons we'll get to in a second. And once it gets there, it has to go through more differentiation because it still has some structural and molecular changes to make. It has undergone some differentiation along the way, so it's more like an immature neuron by the time it reaches its 5th layer fate. But let's look at how differentiation happens. How is it determined what type of neuron our neuroblast is going to be? Generally, because our example is going to be a cortical neuron developed from the telencephalon, those tend to differentiate into pyramidal neurons. And pyramidal neurons are excitatory neurons. So our neuron is going to differentiate into a pyramidal glutamatergic neuron because if you remember from the neuron physiology episode, glutamate is the main excitatory neurotransmitter. And the 5th cortical layer is where lots of glutamatergic pyramidal neurons live. So how does this happen? How does our neuroblast know how to turn into a glutamatergic pyramidal neuron that will live in the 5th cortical layer of the cerebral cortex? Well, neuronal differentiation involves complex external and internal molecular signaling pathways and their influence on gene transcription and expression that happens during migration. This is how I understand it. The DNA sequence of your genome that lives in every nucleus of every cell in your body is like a blueprint to the cellular organization of your whole body. So inside the nucleus of our example neuroblast is this genome blueprint. And our neuroblast is one very tiny part of this blueprint. Even before it became a neuroblast, this cell had already been instructed to become nervous tissue. Its differentiation technically began when it went from being a pluripotent stem cell to a multipotent ectoderm cell. And at that point, it was only going to be able to become a cell in the ectoderm lineage, which is mostly nervous tissue. It next differentiated into a neuroepithelial cell, again part of the ectoderm lineage, but more specifically, nervous system cell lineage. Then it differentiated into a radial glia progenitor cell, and those cells, though still multipotent stem cells, can only differentiate into neurons or glia. It next differentiated into a neuroblast when its radial glia progenitor divided asymmetrically, and our cell got the Notch1 transcription factor nod to migrate toward the peel surface to become part of the newly forming cerebral cortex. Now, throughout its migration and when it lands at its fate in the cortical layer, our neuroblast's genome will be in conversation with extrinsic and intrinsic molecular signals that will instruct the cell how to become its very special part of the blueprint. For instance, neuroblasts that will become glutamatergic neurons, they may induce a specific sequence of transcription factor expression, and this sequence of expression activates some genes and suppresses others in order to regulate the neuroblast's identity, in this case, glutamatergic. It also regulates its structure, pyramidal, and its fate. In our example, that's the 5th cortical layer. Another way to say this is that this sequence of transcription factor expression is instructing the neuroblast how to turn itself into a glutamatergic pyramidal neuron and how it should find its home in the cerebral cortex. That signaling will also instruct the neuron to make the biochemical components that give it its identity, including producing transport vesicles and the enzymes needed to metabolize glutamine into glutamate. When our glutamatergic neuron has differentiated and found its spot in the cortical layer, it will keep differentiating until its axon and dendrites are fully formed, though axons may start sprouting during migration. Differentiation will also continue until the neuron has all the components that make it an excitatory pyramidal glutamatergic neuron, like protein receptors and receptor channels, and all the other biochemical and structural properties it needs to perform its function. And when all the cells of the cortex have found their fate and the 6 cortical layers are fully formed, the radial glia transport processes will retract. Just to reiterate, aside from migration patterns, this is how most of the neurons in the brain develop. They differentiate in similar ways. Extrinsic and intrinsic signaling pathways activate or suppress gene expression that will determine the neuron's identity, structure, and fate in the nervous system. Glial cell differentiation is not so well understood, but there does seem to be an order to differentiation, at least in the central nervous system. Glial differentiation happens after neuron differentiation, except radial glia because those provide the scaffold for neurons and other glia to make their way to the cortex. But radial glia won't remain radial glia. They'll eventually differentiate into either neurons or glia, though there appears to be some radial glia in the developed adult brain. I briefly discussed those in the glia physiology episode. But otherwise, glia differentiate after neurons, and they'll differentiate into astrocytes and then oligodendrocytes. And if you remember, oligodendrocytes are the cells that collectively are known as myelin, and those particular myelinating cells cover the axons in the central nervous system, and myelin isn't fully established until young adulthood in the central nervous system. Neurons in the peripheral nervous system, like motor neurons, also start out as radial glia and follow signaling pathways to differentiate into motor neuron progenitors and then begin differentiating into motor neurons along the way to their fate, or destination, as well as after they get there. I just want to point out that one of the extrinsic protein signaling pathways involved in neuron differentiation and fate, including your motor neurons, is called sonic hedgehog. For real, you have a gene that someone at some point named sonic hedgehog, and that gene provides the instructions to make sonic hedgehog protein that is secreted and acts as a molecular signal to kick off pathways involved in all kinds of neurogenesis business. But I think it's especially fun that our baby motor neurons were brought to life by sonic hedgehog protein juice. Anyway, okay, so all these neurons in glia are proliferating and differentiating and becoming very specific types of neurons and arriving at their destinations within the nervous system. They're aggregating into what will be specific brain regions. And what I mean by that is that neurons of the same type will recognize each other, probably by protein tags. And then they're going to start forming synapses both locally and across regions. But remember, baby's brain has double the neurons it needs, so there will be competition for those synaptic connections, and those that are underused will be eliminated. But we're getting a little ahead of ourselves because this synaptic connection business, though it obviously begins before birth, carries on for a long while after birth, and then in phases throughout childhood. Synaptic connections are establishing as early as 15 to 20 weeks gestation in a process called synaptogenesis. If you remember from the neuron physiology episode, synapses are the connections between neurons, and they involve the terminal end of the axon almost touching either a dendrite or a cell body of the postsynaptic neuron. But there's a microscopic space between them called the synaptic cleft, where chemical molecules diffuse from terminal end to the receptor channels in the postsynaptic neuron membrane, and they will launch an action potential if the signal is strong enough. If you haven't a clue what I'm talking about, the neuron physiology episode will fill you in on all of the details. So for the synapse to form, the axon is going to be guided by molecular signals to grow and navigate its way to its regional fate. Some axons remain local, as in short distances between neurons, as we see in interneurons. But our glutamatergic pyramidal neuron in the 5th cortical layer is going to have a pretty lengthy axon that becomes part of a network of axons that make up the brain's white matter. So maybe our glutamatergic pyramidal neuron's axon is going to extend to a subcortical region like the thalamus. And molecular signals that are involved in the neuroblast's migration are also involved in guiding the axon to grow towards its fate. And in this case, that would be the dendrites of neurons in the thalamus. But get this, some of that guidance is regulated by chemotaxis. So the axon is either attracted to or repelled by certain chemical signals, usually related to the neurotransmitter it will be releasing, even though at this stage it doesn't have the components to synthesize neurotransmitters. It's genetically programmed to recognize its neurotransmitter chemical signature. In our example, the axon is guided to the thalamus by extrinsic and intrinsic molecular signals and transcription factors, but also by glutamatergic chemical signatures attracting it to its final destination. And once it's at the thalamus, a bonkers amount of highly coordinated biochemical and structural business kicks off to produce and assemble all of the components in the presynaptic end of the axon, like synaptic vesicles, calcium ion channels, glutamate, and loads of other things. There could also be chemical transsynaptic communication between the presynaptic axon and the postsynaptic dendrites to find the right connection. And that crosstalk could also guide synaptic assembly. And interestingly, this may be where astrocytes play a role in establishing synaptic connections. This is just part of the work of Dr. Ben Barris and his lab. And if you'd like to know more about him and his legacy, please go have a listen to the glia physiology episode. But relevant to this episode, Dr. Barris and his lab found that astrocytes release protein signals that regulate synapse formation. And to be fair, neurons need all the help they can get. Because if you remember, the terminal end of the axon can have multiple branches of terminal buttons that can synapse to multiple dendritic spines attached to multiple neurons. Needless to say, there are a load of molecular signaling pathways necessary to induce synaptic contact and assembly. It appears, though, to be incredibly efficient and coordinated, and everything the fetus is doing throughout development that is regulated by its nervous system, both voluntary like kicking and involuntary like its heartbeat, all of its wee little actions are stimulating more synaptic connections and strengthening others. And astrocytes appear to have an important role in coordinating those connections. Microglia are another type of glia that may also help with the formation of synaptic connections in the prenatal brain, including dopaminergic connections in the forebrain and interneuron positioning within the layers of the cortex. They may also play important roles in synaptic pruning in the postnatal brain, but we'll get to that in the next episode. If you remember from the glia episode, microglia are macrophages in the central nervous system, and that means one of their main jobs is to survey their surroundings and snack on dead cell debris. They also have a significant role in the immune response. When they detect infection, they activate and induce cell death in pathogens and produce signaling proteins that help control inflammation. But interestingly, they are not born from radial glia precursors like nearly the rest of the central nervous system cells. They don't even seem to generate from ectodermal cells like nearly the rest of the nervous system tissue. They may be born from embryonic macrophage precursors in the yolk sac. We have to go way back to the beginning of the episode to recall that the yolk sac is formed from the hypoblast cells in the bilaminar disc, and it serves to nourish the embryo until the placenta develops and takes over. Yolk sac-derived primitive macrophage cells find their way into the bloodstream once embryonic blood circulation is established. And from there, they find their way to the neuroepithelium, and that's where all the rest of the central nervous system cells are differentiating. These macrophage precursors hop out of circulation and find their way into the neuroepithelium. And similarly to the other cells, specific molecular signals cause these macrophage precursors to express the genomic properties that instruct it to differentiate into microglia. And they'll migrate throughout the developing central nervous system and gobble up neuronal debris. But it's also thought that they participate in remodeling prenatal synaptic connections. But it's not certain how and to what extent. But there is some evidence that microglial processes are attracted to synaptic assembly, possibly by way of chemotaxis. And there they may release chemical and protein signals that influence synaptic assembly or elimination. And there is growing evidence that significant disruption of microglial influence on neuron development and synaptic assembly could result in neurodevelopmental disorders, including autism and schizophrenia. Though more studies are needed before we can make any confident assertions there. There are also a number of studies focusing on fetal alcohol exposure and its detrimental effects on neuron, astrocyte, and synaptic development, and more recently, its relevance to microglia's influence on the developing brain. Broadly, it is thought that it may impede microglia's immune response functions, resulting in widespread pro-inflammatory environments, which is toxic to neurons and may interfere with microglia's roles in synaptic assembly that would normally result in the early establishment of neuronal networks like neurotransmitter pathways. But again, more studies are needed to understand microglia's roles in fetal brain development before we can fully comprehend any interruptions in fetal microglia function. Okay, so let's assume there are no apparent developmental issues with our wee embryo, and hundreds of millions of neurons are migrating and maturing, and some of them have already begun forming connections. And then we have glia migrating and maturing and doing all kinds of business to help the neurons in all kinds of capacities, including guiding them to their destination and helping them form synaptic connections. And the neurons are guided and or attracted to other like neurons so they can form clusters of nuclei and ganglia and brain structures that beget brain regions. And the cortical layers grow, and the cortex will outgrow the skull cavity and begin to form folds and creases called gyri and sulci, respectively. And at birth, though baby's brain has more neurons than it knows what to do with, it hasn't formed all of the synaptic connections yet. It has all the structures it's going to have, but the structures that are mostly online, meaning have established connections, are the brainstem and spinal cord. When baby is born and interacts with its environment and people, it will make many synaptic connections, especially in the cerebral cortex, more than it needs. But that will settle down over time. But we'll get into all of that in the next episode. For the rest of this episode, we're going to look at the role of hormones on fetal neurodevelopment. Though hormones play a role in many areas of fetal development, we're going to look more specifically at the way hormones promote and affect brain development. The first hormones we'll look at fall under a broader group of hormones produced and secreted by the adrenal cortex. Those are glucocorticoids. You may remember glucocorticoids from the nervous system overview episode. Cortisol is an example of a glucocorticoid, but it's not the only glucocorticoid. This group of hormones has broader influences on fetal development, but its role in brain development is regulatory and requires just the right amount to do its business, which involves hippocampal neurogenesis and other neuron anglial proliferation and maturation. Too little will cause developmental issues. Too much will also cause developmental issues. Maternal or parental cortisol is passed on to the fetus through the placenta. So fetal cortisol levels have been shown to be highly correlated to maternal parental cortisol levels. Microglia in particular can become overactivated by overexposure to cortisol in the prenatal brain, which could result in inflammatory responses that disrupt development. Other hormones of note are maternal/parental estradiol and progesterone. These are very important hormones in fetal development. They are also passed through the placenta to the fetus, though progesterone may also be synthesized in fetal adrenal cortices once those organs are able to do so. Estradiol may promote axon growth. It may also influence proliferation of oligodendrocytes and thus have a role in central nervous system myelination. Progesterone is synthesized in a number of brain structures, but it's not certain the precise ways in which it modulates brain development. But it has shown to have neuroprotective effects during development in rodent models. It's also been implicated in neuron differentiation and synaptic connections, oligodendrocyte differentiation and axon myelination, as well as brain sex differentiation, which we'll get to in a moment. Much of the study involving estradiol and progesterone's influence on brain development has been done in animal models, so again, we can't make super confident assertions that any of these things happen in human models. There are other hormonal influences from the thyroid and insulin growth factor, but I'll post a paper in the reference section for you to investigate on your own, because I'd like to devote the remaining minutes of this episode to the genetic and hormonal influence on gonadal differentiation and how that may influence brain sex differentiation. And this is just a fancy way of saying that depending on the embryo's gonads, Some of its brain structures will later undergo masculinization or feminization. And those are phenotypical labels given by scientists to distinguish the structural differences between brains that marinated in androgens in utero and brains that developed in the absence of androgen exposure. But I want to be clear-ish about a couple things up front. One is that the word differentiation in this instance is different from how it's been used up to this point in the episode. This is not about pluripotent or multipotent stem cells morphing into a particular type of cell within one individual. This is about two phenotypically different individuals and the differences between their brain development. The second is that sometimes brain differences are phenotypically distinct, and sometimes they're not. Sometimes there's a lot of overlap and/or small differences between them. From what I can discern from the literature, discussions on brain sex differentiation are based on averages. So to understand what I mean by brain sex differentiation, we have to go all the way back to the beginning of this episode when the sperm cell dropped its haploid nucleus into the oocyte. Remember, that's half of a full chromosomal set. So that sperm haploid only had 22 autosomes and one sex chromosome, either an X chromosome or a Y chromosome. And the oocyte had 22 autosomes and only an X sex chromosome. If the sperm haploid had a Y sex chromosome in its chromosomal set, the resulting zygote would have 44 autosomes plus XY sex chromosomes and will go on to produce testes. If the sperm haploid had an X sex chromosome in its chromosomal set, the resulting zygote would have 44 autosomes plus XX sex chromosomes and would go on to produce ovaries. So typically, to be clear, XY zygotes will go on to grow testes and XX zygotes will go on to grow ovaries. Now there are chromosomal anomalies that can happen, and I explained what some of those are in the cell physiology episode, so I won't go over them again here. But as the zygote matures, whichever set of sex chromosomes the embryo has will determine the embryo's reproductive organs, and those organs may or may not secrete certain hormones that will later partially influence the morphology of some brain structures. So, if our embryo's reproductive organs are testes, it will produce androgens and secrete them into circulation, and those androgens will inform how some of those brain structures will form. If its reproductive organs are ovaries, there will be an absence of androgens, and that absence will inform how some brain structures will form. There are also transcription factors thought to be involved. It's likely not just androgens or the absence of androgens that induces differences between XX and XY reproductive organs and brains. But androgenic influence has been the widely accepted understanding of brain sex differentiation. But let's take a closer look at what's happening. As the embryo develops its reproductive organs, the gonads are bipotential, meaning until they are told to become testes or ovaries, the developing reproductive organs are virtually the same, regardless of XX or XY chromosomal designation. There is a very sensitive window of time during gonadogenesis when sex determination happens. What turns on gonadal determination is either the presence or absence of a gene called SRY. SRY is present in the Y chromosome, and its expression encodes a protein called testis determining factor, or TDF. And TDF kicks off the differentiation of steroidogenic precursor cells. Those are going to produce the androgens in this instance. They also kick off primordial germ cells in the developing gonads, which differentiate into those hardworking Sertoli and Leydig cells, and some of those will differentiate into cells that form the testes. The fetal Leydig cells, those are going to produce androgens and pump them out in abundance. Those androgens will continue to inform the development of the body and then later the brain. In particular, there is a surge of androgens which include testosterone and dihydrotestosterone, or DHT, and those influence the development of genitalia as well as the Wolffian duct that will differentiate into the epididymis, vas deferens, and seminal vesicles. The Sertoli cells secrete anti-Mullerian hormone, which represses the development of the Mullerian duct. The Mullerian duct is the precursor to the XX reproductive tract, which includes the fallopian tubes, uterus, cervix, and the upper vagina. Its regression also allows for the formation of the Wolffian duct. If the embryo does not have a Y chromosome and instead has two X chromosomes, it won't develop testes. There may be some transcription factors in XX genes that actively repress testes formation, but it's not yet certain. It's mostly been thought that if the SRY gene doesn't launch testis-determining factor, then the reproductive organs will just passively become ovaries. So we pretty much don't know entirely how ovaries become ovaries outside of the absence of androgens. And I'll say it now, and remind you again later, we don't seem to know much about the mechanisms that feminize brains either. And ovaries also secrete sex-specific hormones, but not until puberty. So the current understanding is that without exposure to androgens or anti-Mullerian hormone, the ovaries will initiate development and the Mullerian duct will continue to develop because there's nothing stopping it, and it begets most of the XX reproductive organs. Now, gonadal determination occurs around the 6th week of gestation, but brain masculinization or feminization doesn't occur until the last trimester, so beginning somewhere around the 25th week. Until then, the brain in both XX and XY embryos are also bipotential, meaning they can be either masculinized or feminized. And again, all that really means is that some brain areas organize slightly or not so slightly differently based on the presence or absence of prenatal androgen. And though masculinized brains can show childhood proclivity to objects and playstyles and behavior, socially deemed masculine, the theory is that many of these brain differences don't fully manifest until they're activated at puberty, when either testosterone influences the XY brain or estrogen influences the XX brain. Though that theory has mostly been tested in rodents and birds that sing. There are other brain sex differentiation theories involving XX and XY chromosomes and their gene expression. For instance, the SRY gene in the Y chromosome may continue to influence brain differentiation later in gestation, and both chromosomal sets may have genes that encode transcription factors that influence masculinization or feminization of some brain structures. But we don't really know, and that's less to do with hormones, so probably a topic for another day. The distinctions I want to emphasize are first that gonadal determination has a very sensitive window. Any disruption in androgen exposure in XY embryos will kick off a cascade of effects that will affect gonadal differentiation and possibly later brain masculinization, though other transcription factors may offset some of those deficits. We just don't know. Conversely, exposure to androgens in XX embryos during that window also kick off a cascade of effects that impact gonadal development and possibly later both brain masculinization and feminization, or neither. And this is where we close the loop on Dr. Ben Barris' story from the GLIA episode. Dr. Barris' mom was prescribed an androgen-based anti-miscarriage drugs during her first trimester, which would have passed into fetal circulation through the placenta, exposing XX embryonic Ben to androgens. Ben was born without most of his reproductive tract, though he did develop ovaries. He also suffered from gender dysphoria from as early as he could remember until he transitioned to his experienced gender at midlife. And being the consummate neuroscientist that he was curious if his early embryonic exposure to androgens caused a developmental cascade that led to the masculinization of his brain, and perhaps partially explained his proclivity toward masculine-labeled toys and interests throughout his childhood. Though he was quick to acknowledge that neither does one have to be prenatally exposed to androgens to experience gender dysphoria, nor does one require brain masculinization to have an interest in socially deemed masculine playstyles and objects. I mention it because he is an example of an XX embryo who was exposed to androgens during the sensitive gonadal determination window, which may have affected his reproductive tract development and possibly influenced a masculinization cascade. The second distinction I want to emphasize is that gonadal determination is not concurrent with brain masculinization or feminization. There are many weeks and a lot of brain development separating these two processes, meaning any disruption to neurodevelopment in between those events could affect the process of brain masculinization or feminization in the last trimester. There are some theories that suggest disruption to brain masculinization or feminization may be correlated with gender dysphoria and or gender fluidity. This is difficult to disentangle until we have a better understanding of the mechanisms of brain masculinization and feminization, as well as gender dysphoria. But it has me curious if there is a connection between disrupted prenatal neurodevelopment and atypical brain sex differentiation, and how that might influence gender identity. Because earlier last year, a meta-analysis by Kalatsinaki and Williams found a high prevalence of gender dysphoria and gender incongruence among people with autism spectrum disorder. Now, one can be on the spectrum and not experience gender incongruence and dysphoria, and one can experience gender incongruence and dysphoria and not be on the spectrum. But they did find that a statistically significant percentage of ASD people experience gender incongruence and dysphoria. We don't fully understand the etiology of ASD, though it's likely to be multifactorial with genetic and non-genetic implications. We don't fully understand the etiology of gender incongruity or gender dysphoria either, though the research suggests it could also be multifactorial with genetic and non-genetic pathways. And as mentioned, one particular theory involves disruption in brain sex differentiation. But also, we don't know all of the mechanisms involved in brain sex differentiation. So I'm not making any assertions, I'm just expressing curiosity and putting the question out there. ASD affects more than 20 million children worldwide, and a significant portion of those children may develop gender incongruence that could lead to dysphoria. And as we can ascertain from Dr. Barris's story and the growing inventory of research, gender dysphoria can deeply and severely affect child and youth mental health, as well as interfere with daily functioning. So understanding the neurodevelopmental mechanisms and relationship between autism, brain sex differentiation, and gender incongruence and dysphoria could be very helpful in informing clinical protocols for this subset of the ASD community, and possibly even for children who experience gender dysphoria who do not have an ASD diagnosis. Psychiatrist Dr. Aloke Kinojia, who runs the Healthy Gamer YouTube channel, discussed the Kalatsinaki and Williams meta-analysis, as well as proposed clinical outcomes, so that may be of interest to you. I'll post the link in the show notes below. Okay, lovelies, that's it for this episode. I hope I've been able to communicate in these episodes and across this series that the brain and body, their development and function and homeostasis require hundreds of trillions of tiny and not so tiny events and sequences and expressions and connections and secretions and computations and innervations and so many more shuns. It's mind-boggling. And yet, here we are being our very own vessels of cellular and molecular wizardry. In this episode, we got a tiny glimpse into how your very you self began. There are so many other factors we didn't get into, but I hope I was able to convey that even before you entered the world, you were already a unique and complex and nuanced being with your very own genetic makeup that had, and still has, its own unique response to environmental influences that turn on and off gene expression and made new synaptic connections as you grew into your magical baby self. And next episode, we'll look at what happened inside your brain after you entered the world and follow its development all the way to young adulthood. If you're not yet an adult, well, behold your future. Your homework for this week is to have a look at some wonderful scientists who study developmental neuroscience. A few to look out for are Dr. Deboleena Roy. Dr. Roy is a professor of neuroscience and behavioral biology and women's gender and sexuality studies at Emory University. Among her areas of interest that are relevant to today's episode are reproductive neuroendocrinology and molecular biology. Another wonderful neuroscientist to look out for is Dr. Damien Fair. Dr. Fair is a professor at the University of Minnesota and the Red Leaf Endowed Director of the Masonic Institute for the Developing Brain. Among his research interests are neurodevelopmental disorders, including autism and ADHD, and prenatal neuroimaging. And lastly, we have Dr. Shuba Tole. Dr. Tole is a professor of neurobiology at the Tata Institute of Fundamental Research. Her lab explores genetic mechanisms and signaling pathways in the developing forebrain. Particularly relevant to this episode is her lab's research pertaining to signaling pathways in the neuroepithelium that regulate the early development of the cortex, as well as transcription factors involved in the transition from neurogenesis to gliogenesis, mechanisms that control neuron subtypes in the cortical layers, and the development of axonal tracts in the cortex. Dr. Tole's lab has studied many significant molecular pathways involved in the developing brain. I've also posted a video that shows blastocyst formation, and of course, the Dr. Kinojia Healthy Gamer episode that goes through the autism and gender dysphoria paper by Khaled, Sanaki, and Williams. Okay, lovelies, thank you for being here. Again, please check out the STEM organizations in the show notes and support them or others in any way that you are able. I have no relationship with these organizations. I'm just excited about the work they're doing, and I hope that you will be too. If you liked this episode, please click the like and subscribe and notification bell if you haven't already. Please share it with your friends, neighbors, and family, and anyone who you think would benefit from understanding their body a little better. Until next time, take care out there.